Hello students, this is Dr. Anita Raj, your chemistry mentor, welcoming you for another session in Biomolecules. In this session, I shall teach you about the different structures of proteins, okay? Actually, the structure of protein is studied in terms of 4 levels of structural organization called primary, secondary, tertiary and quaternary structures. Actually, each succeeding these structures, levels of organization is more complicated than the previous one, okay? This is more complicated than the previous one and this is more complicated than the secondary, and quaternary structure is more complicated when compared to the tertiary, and each one is a direct consequence of the chemical features of the previous level. Understood? Okay. So now we are going to see the primary structure of protein. Okay. So primary structure refers to the number of sequence of amino acids in polypeptide chain. This is the amino acid, okay. So these amino acids are arranged in a sequence, right? And they are linked together by a peptide bond or a peptide linkage, okay. So in this structure, amino acids are arranged in sequence, and each polypeptide chain in a protein has large number of amino acids. So this is a polypeptide chain, okay. So this is a polypeptide chain, and this polypeptide chain of protein will be having large number of amino acids, and these are linked with each other in a sequence or in a particular or in a specific sequence, okay. So any change in this primary structure, okay, any change in this primary structure or in the sequence of amino acids will be creating a different protein, okay. So actually the amino acids will be linked together in a particular sequence, okay, in a specific sequence by means of a peptide bond. And if suppose if any of the amino acid is removed, and in that place if we are substituting with another amino acid, that will create a different protein. A small change in just one amino acid can drastically alter the properties of the entire protein molecule. For example, when you are taking into consideration hemoglobin, say I have given normal hemoglobin. This is the specific sequential arrangement of amino acids in normal hemoglobin. Okay, the first one is valine, and next one is histidine. This is leucine. This is threonine, proline. Okay, glutamine, lysine. So the next one is, see, we are changing, instead of glutamine, we are substituting with valine. Okay, we are substituting with a valine. So there is a small change. Actually, this hemoglobin acts as an oxygen carrier. Okay, normal hemoglobin, it acts as an oxygen carrier in the blood, which consists of 574 amino acid units. Okay, so the change in just one specific amino acid in the sequence results in a defective hemoglobin found in protein suffering from sickle cell anemia. Okay, so just we are changing only one. We are changing only one amino acid. Okay, so even a simple small change in this amino acid sequence will lead to a defective hemoglobin, which is called a sickle cell anemia. Can you understand? So the entire activity is changing. It is drastically altering the properties. So the second one is the secondary structure. See, actually the arrangement of polypeptide chains obtained as a result of hydrogen bonding is called the secondary structure of proteins. See here, first the primary structure, the amino acids are sequentially arranged or continuously arranged, that is they are completely linked by means of polypeptide chains, okay, polypeptide bonds, okay. See here in secondary structure, the arrangement of polypeptide chains obtained as a result of hydrogen bonding is called the secondary structure. So the secondary structure of protein is of mainly two types. The first one is helix structure and the second one is beta pleated structure, okay, helix structure and beta pleated structure. Actually, this helix structure is suggested by Pauling in 1951. Okay, and he says that the protein consists of amino acid chains coiled into a spiral called helix. See here, it is coiled, is it not? The amino acids are arranged in just like a coil, right? And this is a spiral, coiled into a spiral called helix. This helix can be it can be twisted to right hand side or to the left hand side. The right-handed helix is called the alpha helix and the left-handed helix is called as the beta helix. Actually, it has been found that the alpha helix is more stable arrangement, and we could see that most of the proteins will be having the alpha helical structure. Can you understand? So in alpha helical structure, the polypeptide chains are spirally coiled by intramolecular hydrogen bonding. They are spirally coiled by intramolecular hydrogen bonding called as helix. Okay, and it's seen when all groups of amino acids are larger. So when in a polypeptide chain, when the R groups are found to be larger, then we can have this alpha helical structure pattern. Okay, and moreover, the proteins with this structure are elastic in nature. Actually, the alpha helical structure, if the protein is having a alpha helical structure and they are found to be then they are found to be elastic. Okay, they can be stretched. Okay, and moreover, the C double bond O group of one amino acid is bonded with the fourth NH group. Okay, it is bonded to the fourth NH group, right? And the example for this one is wool fibers. Okay, keratin in hair and nail. Okay, keratin in hair and nail will be having this alpha helical structure. Okay, and the next one is beta pleated structure, that is beta conformation or the pleated sheet structure. Actually, in 1951, again Pauling proposed another conformation for protein called the beta conformation or pleated sheet structure. Actually, in this conformation, polypeptide chains are extended and are laid side by side. See, this is one polypeptide chain, okay, and this is another polypeptide chain. Okay, this is another polypeptide chain. And these polypeptide chains are extended and are laid side by side, and they are held together by intermolecular hydrogen bonding. Okay, in this case, it's the polypeptide chains are linked by means of intermolecular hydrogen bonding, whereas here it is intramolecular hydrogen bonding. Okay, so and then the structure resembles the pleated folds of drapery, and that's why it's having the name beta pleated sheets. Okay, actually there are two possible pleated sheets. The one is parallel, one is a parallel. Okay, one is parallel pleated sheet, and another one is anti-parallel pleated sheet. Okay, anti-parallel pleated sheet. Another one is anti-parallel pleated sheet. See, in parallel pleated sheet, in this structure, all the chains run in the same direction. Okay, all the chains will be running in parallel sheets. All the chains will be running in the same direction. Whereas in anti-parallel sheets, the structures will be alternative. The chains run alternatively in opposite direction. Okay. So the existence of this pleated sheet structure in solid protein is also confirmed by X-ray analysis, and the chains are found parallel. See, we could see this parallel beta pleated sheets in keratin. Okay, in keratin, this keratin will be having the beta pleated sheet, that is parallel beta pleated sheets, whereas in fibronin we have the anti-parallel arrangement. Can you understand? Actually, the tertiary structure of protein refers to the definite geometric pattern, okay, in which the entire protein molecule folds up to produce a specific shape. Meaning, this tertiary protein or the tertiary structure refers to the three-dimensional shape of a protein molecule. Okay, this gives the three-dimensional structure of protein molecule. This type of tertiary structure results from twisting, okay, then bending and folding the helix. Okay, when we are twisting, bending and folding the helix, we'll get this tertiary structure. Okay, the tertiary structure involves the folding of the entire molecule. Actually, we get this secondary structure from the primary structure, and here the tertiary structure we are obtaining by the folding of the secondary structure, and gives rise to two major molecular shapes called fibrous and globular. So in tertiary structure we have two types, that is fibrous and globular. See here, this is the tertiary structure. Here we have both the pleated sheets. Okay, pleated sheet is seen in secondary structure, and the next one is alpha helix. Okay, alpha helix. So due to the twisting, bending and folding of this helical structure, we will be getting the tertiary structure, and also these two get together. Okay, together the entire protein molecules will be folded up to form a specific shape. Can you understand? So the main forces which stabilize the secondary and tertiary structure of proteins are hydrogen bonding, disulfide bonding, okay, and then electrostatic force of attraction, van der Waals forces of attraction. All these different types of forces are involved in this folding process. Can you understand? So as I told you earlier, this tertiary structure have two types, that is fibrous and globular. So let us see what is fibrous and globular now. Okay. Now let us see what is fibrous protein. Actually, this protein consists of a thread-like molecule. Okay, this fibrous will be having thread-like molecule, whereas this globular In these proteins, the polypeptide chains coiled up and gives a spherical shape. So this globular will be giving a spherical shape, whereas this fibrous will be having a thread-like molecule. And here these are held together by means of disulfide bonds and hydrogen bonds, whereas here intermolecular hydrogen bonding are involved and they are very weak in globular bonding. Okay, and this fibrous protein, they are insoluble in water. This fibrous protein is insoluble in water, whereas this globular protein is soluble in water. Okay, and see here this fibrous protein, they are very stable to moderate changes to temperature and pH. So they are not very much affected by a change in pH or in change in temperature. Whereas here these globular proteins are very sensitive to the temperature change and pH change. Okay, and example for this fibrous one is keratin in hair, nails, then myosin in muscle, then fibronin in silk, wool, etc. are examples for this fibrous protein. Whereas globular, fibrinogen, albumin in egg, hemoglobin, then all types of enzymes, okay, and then antibodies, then venom of snake and scorpion, all these contains this globular protein. Can you understand? So the final one is the quaternary structure. Actually, it refers to the determination of the number of subunits and the spatial arrangement of these subunits with respect to each other in an aggregate protein, which is called as the quaternary structure. Okay, so the spatial arrangement of subunits, how many number of subunits are present, as well as the spatial arrangement of these subunits with respect to each other in an aggregate protein is called as the quaternary structure. And these proteins are composed of two or more polypeptide chains referred to as subunits. Okay, so most of the proteins are that is the quaternary structure of proteins are composed of two or more polypeptide chains which are referred to as subunits. Okay, what do you mean by subunits? If the subunits will be having two or more polypeptide chains. Okay, the spatial arrangement of these subunits with respect to each other is known as the quaternary structure. Can you understand? Well, fine students, I hope you might have understood what I have taught today. Let me meet you with another important topic in my next session. Until then, it's Dr. Anita Raj, your chemistry mentor signing off from you. Thanks for watching.